This video, I am going to talk about nucleotide excision repair. So this is a DNA repair process that specializes in repairing structural damage in DNA. So what do I mean by structural damage? So not double strand breaks, right? We have a different repair process for that. We covered homologous recombination based double strand break repair in previous videos. So a type of structural damage that nucleotide excision repair, also called NER, is in charge of is, let's say, cyclobutane pyrimidine dimers. Now these are also called CPDs. So what are cyclobutane pyrimidine dimers, CPDs? So by now we should be very familiar with the nucleotides C, T, A, and G, C, and T. These are the pyrimidines, and these are purines. So CPDs involve the pyrimidines. So, and they occur when we have two pyrimidines side by side in a strand of DNA. So DNA is double-stranded, so if we should take one strand and find, let's say, a C followed by a T, it could be the other way around with the T at the 5' end followed by a C. We could also have two T's or two C's. So I can also refer to these as dinucleotides. So anywhere along the DNA molecule, if there are two pyrimidines next to each other in the strand of DNA, these have the potential to form a CPD. Now CPDs tend to form when DNA is exposed to UV light. So UV light causes the rearrangement of electrons in the nitrogenous bases of these nucleotides that causes the formation of that cyclobutane pyrimidine dimer. Now, let me look at the structure with you a little bit so you can see how this works. So let's take a look at the structure of thymine, just the nitrogenous base. So the ring structure, remember pyrimidines have a single ring in the structure. There's an end there, and then this is the end that links to the sugar, the glycosidic bond to the sugar. And there's a methyl group right here. So this is the structure of thymine, and let's take a look at the structure of cytosine too. So they're very similar. And okay, so how are these different? You know, we have a double bond here, no double bond there. We have a methyl group here, CH3. That's a funny way to write that, right? No methyl group here. We have a carbonyl group here. And, oh, maybe I should call this a ketone. I forget my organic chemistry. And an amino group here. We have a hydrogen here. And here, there should be a double bond right there. Okay, so when we have these together, whether it be two T's, a T and a C, or two C's, or a C and a T, side by side in a DNA molecule, what can happen is we can get a structure like this formed. So let's focus on two T's. So I'm going to flip this one around relative to how I had it in the previous diagram. Let's see here. So here is where we're going to get the cyclobutane start to be formed, right here. So this is a normal thymine nitrogenous base right here. And what I'm doing is I'm extending these two carbons this way, and they connect to the other thymine in a thymine dimer or a thymine-based CPD. Okay, so there should be an N here. Okay, so it looks something like this. Now, you know, this is the cyclobutane. So butane, I think, is for 4 carbons, right? So we have 1, 2, 3, 4, and it's cyclo is a circle here. So this is a cyclobutane pyrimidine dimer. You know, we could easily see how we could change this structure to a C to make this a TC CPD. But we'll just focus on this one. So, okay, so why are these bad? And you know, it doesn't seem like it causes that much of a problem, right? Oh, at least when I diagram it like this. Well, let's take a look. So I have a diagram here I think we can use to speed things up a little bit. So let's say this is a random interval in a DNA molecule. The DNA molecule is much longer. These squares represent the 5 carbon sugars. I indicated the anti-parallelness of the 2 strands. We have the bases here and here. We can see 3 hydrogen bonds forming between the GC base pairs and 2 hydrogen bonds between the AT base pairs. Now, okay, so let's say UV light should hit this region right here and cause a CPD to form between these 2 T's right here. Well, without worrying too much about exactly, you know, the exact structure and lengths of the bonds and things like that, conceptually what happens is we get these covalent bonds formed between this molecule, and that changes the distance here in the rotation of the thymine nitrogenous bases here relative to how they look when they're not in this dimer formation. Now that screws up a lot. So changing the distance between here, I don't know if it gets greater or smaller than what it is when this cyclobutane is not here, but we changing the distance between these 2 sugars and these 2 nitrogenous bases. We're interfering with the hydrogen bonding with these A's right here, and it's often said that it causes a kink in the DNA. So researchers who study structure will tell us that, okay, when this forms, we get this kink in the DNA that shouldn't be there. And that's the structural damage. So you can imagine it probably throws off all the base pairing in this region of the DNA molecule also. So this is the structural damage that must be repaired. Now, if it's not repaired, what do you think would happen? So let's say it's not repaired and this section of the genome needs to be replicated, say during S phase. So this template right here, if this was the template, the top strand, everything would probably be okay. But if this bottom one was used as the template by DNA polymerase, well, it's probably going to have a hard time replicating this DNA accurately, right? Because the base pairing, proper base pairing can't occur when you have a thymine dimer in the template. Additionally, RNA polymerase if it should need to use this, if this is the template strand of a gene that needs to be transcribed by RNA polymerase. RNA polymerase can't bypass, I'm not sure it can read it, but it can't bypass this thymine dimer. It stalls. And so it can't be transcribed. And as we'll see, that is one of the ways the cell can tell that there's a thymine dimer, is when a gene's trying to be transcribed, RNA polymerase stalls at the dimers, at the CPDs, and initiates nucleotide excision repair. So that's one of the detection processes. So the cell has ways to fix these, and it's pretty important that it does. And the major way to repair those is nucleotide excision repair. And this is, a critical, maybe all the repair processes are critical, but this one seems to be especially critical in humans. And, probably other mammals too, and maybe even other eukaryotes, compared to other DNA repair processes. And the reason I want to say that is we know of a pretty severe genetic disease, xeroderma pigmentosum. And if you have this disorder, it's because you lack one of the proteins needed to perform nucleotide excision repair. So your cells cannot perform nucleotide excision repair. So your cells can't fix the CPDs. As a result, you really can't go out in the sun. So no sun exposure. And even when they're doing their best to stay out of the sun, cancer rates are still super high. And I'm not sure if this is for patients who never ever have been out in the sun, or if it's even if you have XB and you avoid sun. But cancer rates are much, much higher, something like 1,000 times to 10,000 times higher than a human without this disorder. Skin cancer rates, right? So humans really need nucleotide excision repair to spend any time in the sun at all. So we go in the sun, we're going to have some CPDs. Those CPDs need to be fixed by nucleotide excision repair. So let's take a look at the mechanism. How does it work? So nucleotide excision repair can be split into two distinct mechanisms. Well, connected mechanisms. They start out differently. One is called global genomic repair, and the other is called transcription coupled NER. So, well, I should say global genomic nucleotide excision repair and transcription coupled, or coupled, not coupled, NER. And these two pathways differ by the way the structural damage is detected. So over here, the detection is going to involve one of two complexes. So one protein complex contains two proteins, XPE and DDB2. You can probably guess where the XPE came from researchers who discovered these proteins while studying xeroderma pigmentosum, or the causes of xeroderma pigmentosum. And the other complex that can detect these CPDs is XPCHHR23B. Now over here, two of the proteins involved in the detection process are going to be called CSA and CSB. Now these might have roles in, well, RNA polymerase 2 is going to be a major component of detection or detecting the CPDs. But these help with the process. So these two proteins right here are specific to TCNER, and these over here are specific to GGNER. And it's thought that these might work. We're not exactly sure how they're working, but they might work by helping RNA polymerase to maybe moving it off the template or adjusting RNA polymerase on the template when it stalls, or adjusting it in a way that allows the pyrimidine dimer to be repaired. I said RNA polymerase 2, and we haven't really talked about what the various RNA polymerases do, but RNA polymerase 2 is the RNA polymerase in charge of transcribing mRNA encoding genes. And TCNER works with RNA polymerase 2. I'm not really sure about the others. It's okay if we just focus on RNA polymerase 2. RNA polymerase 1 and RNA polymerase 3 are the others. Okay, so let's start with GGNER and see what happens. Now I've got some little cutouts of some proteins and DNA molecules. Let's see how this works. So let's assume this is a thymine dimer in a DNA molecule. So we have the recognition complexes that will search the DNA in the genome for CPDs. And this CPD is a thymine dimer. And only one of these has to detect it, but I have two here. Why not? XPE and DDB2. That's one complex. The other complex is XPC and HHR23B. So one of these will detect the CPD. And when it detects it, it will recruit a transcription factor called TF2H. Now we will see in future lectures that TF2H is a key transcription factor with respect to initiation of RNA transcription. And it has, it's comprised of several proteins. Two of them are XPB and XPD. Now these are helicases, and they are thought to unwind the DNA that contains the pyrimidine dimer. So about 15 bases, making a bubble, let's say, around the pyrimidine dimer. And I was going to say about 15 bases on each side, and it might be a little longer than that. So I'm going to replace that. Okay, we have our unwound DNA with the pyrimidine dimer, and I'm going to put those protein complexes back in there. Now, almost any time we see an unwound DNA with single strands, we can bet what protein is going to be involved. We've seen this many times. Replication protein A. So we're going to put a couple of these in here on the single strands of DNA. And it's pretty safe to say that, what does RPA do? Well, binds single-stranded DNA. And why is that important? Well, maybe it helps keeps it from coming back together. After the TF2H did all that work unwinding the DNA, if they came back together, that would, ruin everything. So if the single strands came back together, that would be bad. I think that's enough. So another protein comes in, and this one we're not really sure what it does. This is called XPA. We know it's important, and some models suggest it's important for making sure that there was damage here to begin with, confirming the damage. Okay, so now the next step is some endonucleases come in. Now we have two endonucleases. A complex containing XPF and ERCC1 is one endonuclease, and the other one is called XPG. Now, they are going to be found on either side of this little bubble that's found that the pyrimidine dimer is located in. And you know what? I'm going to take the detection proteins out because those are unnecessary now, and they probably leave when these nucleases come in. So these guys go one to each side, and each one is responsible for cutting the strand containing the CPD. So one cuts upstream of the CPD, the other one cuts downstream of the CPD. And then what comes next is after it cuts that, so we're going to see that top strand come off. So I can rearrange this a little bit. Now that strand, which is 25 to 30 nucleotides long, is going to come off in a complex with the TF2H transcription factor. Probably going to be a couple RPAs in there. Going to be some RPAs up there still though. RPA binding single-stranded DNAs, right? And XPA is probably gone too. Let me just check to make sure that's the model we're working with. And those two are coming off too, the nucleases are coming off too. And this little DNA now that was excised is called a SED, for small excised damage containing DNA. Small excised damage containing DNA. So a SED DNA. Now, at this point up here, I'm looking at the notes that I had for you guys in class, and for some reason I have XPG still here. Now I can't remember if that should really be XPA or something. It doesn't really matter. I think you see exactly, we know, we can see what's going on with this mechanism. So, but at this point, what's going to happen? So DNA polymerase is going to come in, and it's going to be loaded on with pCNA. Now these are enzymes and proteins that we've seen during our lecture and discussion of replication. So replication factor C is important for loading pCNA on with DNA polymerase. So these come in, and DNA polymerase now what polymerase? So current models suggest it's delta, epsilon, or another one called kappa. And what these can do is, you can use the 3' OH right here to synthesize DNA, using this strand right here, the good strand that wasn't damaged, as the template. So it's going to synthesize all the way up here, and what it's going to leave is one nick. And I didn't even make, a little shape for the next enzyme that's going to seal this nick, because I know you guys already know what does that. So DNA ligase. So one of the DNA ligases in the eukaryotic cell will come in and seal that nick. Okay, so that is GGNER. So what about TCNER, transcription coupled NER? So let's assume that, we can go back to the very beginning. There's a CPD here. Now let's say this is part of a gene. This DNA interval here is found within a messenger RNA encoding gene. And before GGNER finds this damage, what happens is, the cell tries to transcribe this gene. So, and we haven't talked about transcription in detail yet, but I'll just talk about the very basic stuff that you've learned in previous biology courses. RNA polymerase is going to use this top strand right here as the template, if it's using the top strand as the template, and this little red squiggly thing coming out of here, that's the nascent RNA that's being transcribed as RNA polymerase is moving down the DNA molecule, transcribing RNA. And when it hits the pyrimidine dimer, it stalls. It stalls. And these CSA and CSB proteins, they're thought to travel with, well, one model suggests that they travel with RNA polymerase 2. And sometimes, at least at the very beginning of transcription, TF2H is also traveling with RNA polymerase 2. So RNA polymerase 2 stalls at the CPD, and you can see in cases where TF2H is already with RNA polymerase 2, well, the cell is really close to being able to fix that CPD, right? It already has a central component of the machinery needed to perform nucleotide excision repair. So CSB and CSA are important for getting RNA polymerase 2 out of the way. At least we think they're important for getting RNA polymerase 2 out of the way and allowing TF2H in there. And then the nucleotide excision process can continue, based on, where TF2H enters the process in GGNER. So the pathways converge when TF2H takes over for RNA polymerase 2. I guess I could go over it quickly. So we're going to have RPA in here. And I lost my XPA. XPA in here. So, okay, so at this point, that's when the nucleases come in. Where's my other nuclease? Here it is here. Okay, so now you can see that SED is going to be released. We're going to lose TF2H, XPA, the nucleases, and can't help myself. DNA polymerase can come in with replication factor C. Fill in the damage. And then we need DNA ligase to seal that nick. So that's it really for nucleotide excision repair. Not too complicated, I don't think. I hope you enjoyed the little, I don't know what I would call these, cutouts I made of the various proteins. I tried to keep the colors consistent with what we have in the lecture notes. So I hope that helps you understand nucleotide excision repair. That was kind of fun putting that together. And let me know if you have any questions. Make sure to check out the questions in the notes, and I'll see you in the next video.